Devania Cheek, and I will be discussing my group's project proposal on investigating the effects of microgravity on plant genetic transformation. Oh, sorry. For a little introduction, genetic engineering is an emerging prospective method for evaluating, for optimizing space agriculture by selecting for favorable traits in plants for these conditions. Agrobacterium is, Agrobacterium is a bacteria that is known for horizontal gene transfer. This proposal is looking at using Agrobacterium tumor fasciitis to conduct this horizontal gene transfer in microgravity. For a little background, Agrobacterium tumor fasciitis is a gram-negative pathogenic bacteria to plants, and it is able to reprogram host cells by injecting their own DNA inside of these cells. They contain a plasmid that contains a fluorescent protein as well as a gene that codes for an antibiotic resistance. The gene will serve to allow for the gene as well as the fluorescent protein will serve as a selection for determining which seeds actually went through the transformation process. As for the experimental design, it will involve a tube with 3 volumes and 2 clamps separating the volumes. The first volume will contain a dehydrated Agrobacterium and tobacco seeds. The second volume will include a hydration growth medium solution, and the third volume will contain the antibiotic kinemycin. First, the first clamp will be removed to rehydrate the bacteria and allow for this horizontal gene transformation process to occur. And after a sufficient amount of time, the second clamp will be released to neutralize the bacteria to prevent any further damage to the seed. And the seeds would be brought back to examine how successful this horizontal transformation was under microgravity. Some questions or points to be addressed will be the efficiency of the transformation method of conjugation in microgravity, as well as insights on optimizing space agriculture and plant host interactions and genetic targets for regulating infection, as well as overall just improving genetic engineering in plants. I would like to acknowledge my co-investigators, Rahi Kajastar and Eva Burnell, as well as our professional advisors, Dr. Andrew Palmer and Dr. Armando Azu Batista, for their expertise in microbiology and plant physiology.
Thank you very much. Do we have any questions?
I actually have a question. What do you think is potentially going to be the difference between an Earth-based experiment versus the experiment in microgravity? And just for completeness, I just wanted to say that all of the Mission 19 and 20 experiments that are being presented today have not flown yet. So the most that they can do is optimize their experiment for microgravity and hypothesize what they think might occur or might not occur on orbit relative to the ground control.
I think that, well, we think that this process will be a bit more successful given the increase in virulence for viruses and bacteria in space and microgravity. So it will have a better outcome for transformation.
Any other questions? All right, thank you very much.